Thank you very much for this comprehensive lecture. I think you have used all your time.
Yes, I'll use all my time, yes.
And we have to move on to the next presentation.
Professor Chung, Professor of Respiratory Medicine, National Heart and Lung Institute, Imperial College, London, Clinical Phenotypes of Severe Asthma.
Yes, thank you. Good morning. Thank you, Professor Campani and Professor Simoni, and also Leo for inviting me again this year to Modena this time. So it's a pleasure for me to talk to you about phenotypes of severe asthma, and you will see that I've dropped the term clinical, because I think when we talk about phenotypes, we need to talk about the phenotyping from the clinical angle and also from the mechanistic angle. I think this is very important, both to the clinician and to researchers like us who are trying to find new treatments for these conditions, particularly severe asthma, where there's a dearth of new effective treatments. I'm sorry, my conflict of interest dropped off, but the usual conflict of interest of attending advisory boards and taking part in various clinical trials and lecturing. So I think it's important to just remind you that what we're talking about. So what is a disease phenotype? So these the definition from the Oxford Dictionary talks about the observable characteristics of an organism which develops with the interaction of the organism in its environment. This, sorry, where's the pointer? There's no pointer. Okay. So you can use airway physiology, inflammatory parameters, body size, and so on to try and describe this phenotype. And the last point is very important. It's a useful but entirely artificial construct to underlying disease entity, i.e., this is a man-made thing. So we construct a phenotype according to observations, according to studies, and who knows what is the the proper disease phenotype? But the value of a phenotype would be whether it gives to the clinician some idea about outcomes of the disease, whether it gives to the researcher some idea about the mechanisms underlying the disease. So I think it's important to realize why we are doing this type of exercise. So there are various ways in which we can do phenotypes, and that mainly it is dependent on what are the characteristics that you put into the analysis. So you can see here that there are three stages of phenotyping in my mind. And initially, a sort of very clinical, clinician-led type phenotyping, the clinico-functional phenotypes, where you use clinical, physiologic, and some biologic hereditary characteristics to build up your phenotype. And then now with the advent of omics technology, where we can measure proteins and metabolites, genes, and so on, you can add this to your phenotyping, and of course this brings on a level of complexity of using mathematical approaches to get to your molecular phenotypes. And finally, the idea of the endotype, which is not different from molecular phenotypes. It's just that then you include some of the molecular mechanisms which contribute to or causes the clinical characteristics associated with the molecular phenotypes. So. And all this is on the basis of this idea of precision medicine, which President Obama talked about last year at the beginning of his presidential year lecture. And so I think this is important. So the treatments are targeted to the needs of the individual patients, on the basis of genetic, biomarker, phenotypic, and psychosocial characteristics, distinguish a given patient from other patients with similar clinical presentation. So what are the characteristics of severe asthma phenotypes? And I've listed this here because I think people will recognize this if you're looking at after patients with difficult to control asthma, or some of them will have severe asthma. So there are some characteristics that are that will be familiar to you. And the early onset asthma versus the late onset asthma, and we know that there may be some different outcomes, whether you have early onset or late onset. The obese versus non-obese, which is now gathering a bit more understanding. The chronic airflow obstruction, and this is obviously linking up with COPD. Recurrent exacerbation, occasional exacerbation, which also links up with COPD. And then the atopic high IG, eosinophilic versus non-eosinophilic, the Th2 dominant, non-Th2 dominant, which is a much more recent approach, a molecular phenotyping, and steroid insensitive versus steroid sensitive, and the poor response to beta-adrenergic bronchodilation versus bronchodilation versus poor bronchodilator response, which I think is an increasingly recognized in severe asthma as it has been recognized in COPD. And I just wanted to mention this very important paper written by Augusti and various other distinguished colleagues in the ERJ just very recently, which talks about a complete new approach and not talk about those very well disease entities that we are familiar with of COPD and asthma, but talk about chronic airways disease and then approach the chronic airways disease in terms of identifying treatable traits. And I think that's a very good approach, which goes into the idea of looking at diseases in terms of different phenotypes. I won't go into this, but it's a very important point. So how do we approach phenotyping of asthma? So as I've said, you start with clinical, functional, and physiological features, and I will show you, of course, some of the more recent phenotyping in that respect. Then we've had to look at whether when we add inflammatory features, mainly from sputum cell analysis, we've been able to refine the phenotyping, particularly in terms of what sort of treatments the patient will respond. And then finally, which is very much still experimental in some way, is to start with gene expression and then associate proteins and with the inflammatory features and so on, and then see what type of clinical and physiological phenotypes you get out of this. So I will try in the remaining time to show you some examples of the phenotypes that we are unraveling in this. So this is in fact the first mathematically driven approach to phenotyping using clustering techniques, and what is shown in this slide is what we call a tree analysis that explains 80% of those patients that were put in this analysis. And you will observe that those patients were a mixture of severe and non-severe asthmatics. And for that sort of exercise, you can't really just look at severe asthmatics in my mind. But you will see that on the basis of four features of the asthma, mainly dependent on lung function, the baseline FEV1, and the bronchodilator response to FEV1, and the age of onset of the disease, you're getting 5 distinct clusters with very little overlap between those clusters. And what those clusters are telling you is an idea of the severity of the asthma. And you will see that clusters 4 and 5 far to your right there, which is really defined on the basis of airflow obstruction with a reduction in FEV1 and a poor bronchodilator response to beta agonist. You define 80% of patients with severe asthma, the cluster 4 and 5. And these are the patients that actually have a reduced FEV1, have more than 3 OCS bursts, oral corticosteroids, and or also on oral corticosteroid therapy, and the typical severe asthma patient. So there's been some usefulness in doing this type of analysis. Now, on the basis of what we know now about asthma, I think this slide from Sally Wenzel summarizes the sort of molecular phenotype that we distinguish now. We distinguish now the Th2 high inflammation versus the non-Th2 high or non-Th2-like inflammation, a Th2 low. And she has tentatively put down some of the clinical phenotypes that underlie those two types of molecular phenotypes. Now, I just want to show you some of the European data from the UBIPRED cohort, which some of you will know has just been completed recently. And this slide really shows the sort of severity of the asthmatics that we have recruited here. In the first two columns here, you can see the severe asthma non-smoking and the severe asthma smoking and ex-smoking. And I think this is a new feature in this study, which is including smokers, smoking asthmatics, and seeing how that is different from the non-smoking asthmatic. And also the severity of the disease of those patients is much more severe than those in SARP. And in SARP, indeed, there was a lot of more obese asthmatic patients, as there's, not surprisingly, as there's more obesity in the States. Now, we did an unsupervised clustering on clinical features in UBIPRED. And I think the idea was, you know, would this be useful to the clinician who sees the patient and who's able easily to measure those features that we've used here into the analysis? The age of onset of asthma, the smoking, the body mass index, measures of lung function, asthma control questionnaire, exacerbations, and use of medication, particularly oral corticosteroid daily use. So I'm not going to go into the intricacies of that analysis, which I believe is a very strong type of analysis. But we came up to 4 clusters. I think some of the clusters shown here are quite similar to what's been described with SARP. And you will see immediately that the cluster of the mild to moderate severe asthma clusters in cluster 1. So this clustering indeed does distinguish between moderate to severe asthma and severe asthma. And the severe asthma clusters 2, 3, and 4 are clusters of severe asthma, but they have other features that distinguish them, and particularly of importance is the difference between cluster 2 and 3, which is a cluster of airflow obstruction. So airflow obstruction comes into cluster 2 and 3, but the difference between them is that there's a highly intensive smoking group of cluster 2, when the cluster 3 is a non-smoking group in general, and then the oral corticosteroid dependence in cluster 3 in the non-smoking group. So, and cluster 4 is an interesting one, which has also been described before, of female obese ladies with frequent exacerbations. So this is, to me, is a very useful classification of patients with severe asthma, and highlight some of the characteristics, how some of the characteristics come together in those clusters. So this is just a detail of the characteristics. The first 7 or 8 lines there I've already shown you in that clustering, but also interesting to see how the clustering, how the data comes up in terms of some of the parameters, of some of the biomarker parameters, particularly in the sputum eosinophils and the sputum neutrophils, as shown here, with the sputum eosinophils, in fact, being much more higher in the group of moderate, well-controlled asthma. So, I think there's been a long interest in terms of whether the sputum eosinophils can help us in terms of the clustering of patients with severe asthma. And shown here is a distribution of neutrophilic and eosinophilic inflammation in the sputum, and the definition of the eosinophilic and neutrophilic inflammation is given here. And you can see that if we compare severe asthma, the non-smoking A versus the mild-moderate asthma, the differences, in fact, that you can see is a greater prevalence of eosinophilic asthma, and also a similar amount of neutrophilic asthma, in 14 and 15%, which has in fact been described before, this type of prevalence of neutrophilic asthma, and a greater increase of the combined neutrophilic and eosinophilic asthma. Indeed, interestingly, in the smokers, in fact, there's in fact less neutrophilic asthma that we see in our severe smokers. But the question is, what value is all this information to us? Well, I think this study in Leicester, which is a classic study, which is, this slide must be shown in every asthma talk on the planet. And I think it really shows the important clusters in terms of the eosinophil inflammation. As you can see, this is on the x-axis, and the symptoms on the y-axis. So you will see inflammation predominant asthma, asthma with the eosinophil, but with little symptoms, and the other way around, particularly the obese non-eosinophilic patient, but with a lot of symptoms. And of course, there is concordant disease where the eosinophil count are those that relates to symptoms. So the molecular phenotyping I've already talked to you about, and just to put you into the picture how this is done, and this is a classic work from Woodruff, where you obtain a transcriptomic analysis of the epithelial brushings, looking for the expression of Th2 cytokines. And you can see there the analysis with those that have high expression of the Th2 cytokines and those that have low expression of Th2 cytokines. And when you look at the features between those two groups, the features of Th2 high asthma are those that have more eosinophilic asthma, increase in serum IgE, increase in mucin MEC5AC, and increase in bronchoresponsiveness, and more importantly, a very good response to ICS, inhaled corticosteroids, which contrasts to the Th2 low asthma that had a very poor response, in fact no response, in FEV1 increase, in FEV1 to inhaled corticosteroids. So that molecular phenotyping distinguished two very distinct characteristics of asthma, two different types of asthma. So I show this now because, of course, there's not only Th2 asthma. Now we should call this T2 asthma, as shown there, but there's also the non-T2 asthma, which we still do not understand the mechanisms. And of course, not shown on this slide, and I wouldn't be able to show it because there are mechanisms that are involved in asthma for which we do not understand, we do not know yet now. And I think what I'm going to show you in the remaining few minutes is some of the phenotyping that we've done at the molecular basis, which will allow us now to try and understand the interaction between those that are on top there, those what I would call the causes of asthma, like allergens, viruses, particulate matter that we inhale in pollution, in pollutants, and oxidants, and how that link up to the characteristics of severe asthma. So I'll pass on this because I'll talk about this tomorrow. But just to remind you that one of the advantages of having done all this phenotyping is to be able to characterize patients that will respond to particular specific therapies, and here this is a therapy directed towards a T2 cytokine, IL-5, and you know that there's mepolizumab that's going to come on the market, in which is directed specifically to this particular phenotype of exacerbating patient with high-dose inhaled corticosteroids and with the blood eosinophil count higher than 150 per microliter. So this is the last part of my talk because I think this is just showing us where the phenotyping is going to go. So when we look at just the genes that are expressed in the sputum cells between severe asthma and non-asthma, or severe asthma and mild-moderate asthma, one of the things that you see here is an increase in those genes that are related to the inflammasome activation. So that's nice data, but what does it mean? And I think we get much more data when we have a much more intensive analysis of this data. And so this data now is what we've done with the data now is to look at the omics, the gene expression, and try and cluster on a gene expression. So you start with a gene expression. You don't start with any symptoms or measurements of eosinophils and so on, but just start with the omics, cluster the omics, and then see what it gives you in terms of the clinical phenotype. So this is exactly what we've done. And here we've taken a hypothesis that there must be differences between eosinophilic inflammation and non-eosinophilic inflammation. So we've looked at the differentially expressed genes between those three groups of subjects with eosinophilic asthma, non-eosinophilic inflammation, and the healthy controls. And we come up to 478 differentially expressed genes. And during the hierarchical clustering, we are now getting, three molecular phenotypes, and we call them DCC1, DCC2, DCC3, disease-driving clusters, because we believe the underlying expression there must tell us about mechanisms. So having done this hierarchical clustering then, so we got three groups, and this is the clinical inflammatory characteristics of DCC1, DCC2, and DCC3, compared to controls, the control non-asthmatic controls. And you will see DCC1 is predominantly eosinophilic, DCC2 is predominantly neutrophilic, and DCC1 also is predominantly obstructive compared to the others. There's some measurements of FeNO here which goes with the eosinophil sputum count. So very quickly, what is this DCC1 looks like? Well, DCC1 is what a prototypic so-called T2 high, but it is mainly characterized by TSLP receptor, IL-33 receptor, and IL-3 receptor, and so it looks like a T2. DCC2 is predominantly pattern recognition receptors, and this is the inflammasome activation. And this is associated, you may remember, with the neutrophilic response. And while DCC3 is associated with a lot of ubiquitination enzymes, and some of them related to mitochondrial enzymes. But we get a much better picture of this, of what those three DCCs means. Here, I'm sorry, it's called TAC, but it's the same. TAC and DCC is the same. And using a technique of GSVA, where we take various signatures, published signatures, and see how much is expressed in those patients. Each of those dots represent one patient in each of those three DCCs. And you will see I've summarized here on the left-hand side what are the characteristics of those DCC1 in terms of the gene expression. So as I said, DCC1 is related to T2 with ILC2 activation, TSLP receptor, IL-33 receptor. DCC2 an inflammasome, neutrophilic type inflammation. DCC3 with TH17, mitochondrial oxidative stress, and aging genes. So DCC3 looks like an aging type molecular phenotype associated with mitochondrial oxidation. And this is just showing the relationship of the sputum inflammation, eosinophilic, mixed, neutrophilic, and postgranulocytic in terms of those DCC1, DCC2, and DCC3. And you can see that the sputum cell count, inflammatory count, doesn't actually allow you to tell you exactly what is which of the DCC1, DCC2, and DCC3 is, and you can see the eosinophilic one is not only the DCC1, but also DCC3. So we're gaining an advantage in doing this molecular phenotyping. So I should just mention that, as Professor Vejiska said, there's a question of time, and on one of the slides I showed time, and I think in terms of the phenotyping, it is important to be doing this phenotyping with time, because we don't know whether there's an evolution and how stable those are. But let me just conclude on this slide, is that I think the phenotyping on clinical parameters may provide useful tools to predict long-term outcomes. The biomarkers may also, the biomarkers that can derive from those may also be useful to determine response to specific treatments. I haven't had time to talk about this particularly. And ultimately, molecular phenotyping and ultimately endotyping will provide mechanistic information and targets for new treatments. And I think you can see that those DCCs that have been described here may link up to some of those causes. And you might think, we think that the DCC2 is related to microbial, particularly bacterial activation that occurs there. And of course, we now know that in severe asthma, some of the work that we've done and others have done in severe asthma, the microbiome in the airways is quite abnormal. And the DCC3 is probably related to exposure to environmental oxidants, such as exposure to environmental pollution, such as car pollution. But of course, we need much more work to try and link up those causes to those DCCs or disease-driving clusters. So finally, the phenotyping and endotyping, I think, are an essential prerequisite for achieving precision medicine. So thank you very much for your attention.
Thank you.